Hey everyone, welcome to my YouTube channel. Let's get started with our today's class 12th biology chapter sexual reproduction in flowering plants. Let's get started. Firstly, let me tell you that flowering plants are known as angiosperms. Without flowers, there will be no fertilization in plants. Let's discuss the parts of flower. This long tube, as you can see, this tube is known as pollen tube or we call it as style. At the top of this tube, this structure is known as stigma. Here pollen grains are stored. At this bilobe structure is known as anther. Below anther, this long stalk-like structure is known as filament. Now you can see below pollen tube this bulged part. This bulged part is known as ovary, and outside ovary this area is known as nectiferous area. In ovary, ovules are kept. Here you can see this leaf-like structure is known as sepal and this is known as petal of the flower. Petal or sepal of the flower, these are the non-essential parts, right? Whereas stigma, style and ovary, these are the female reproductive parts and they are together known as pistil or carpel, right? Pistil or carpel and anther and filament, they are the male reproductive parts and they are together known as stamen, and these are the essential parts for reproduction, means these are included in reproduction. Without these, reproduction will not take place. Pistil are the female parts. It consists of stigma, style and ovary, whereas stamen consists of anther and filament. These are the female parts and are known as gynoecium. Anther and filament are the male parts. They are known as androecium. Androecium, as I've told you, that it consists of walls of stamen. Stamen means anther and filament together are known as stamen. They are the male parts. Stamen consists of filament, that is the long slender stalk, and anther is the bilobed structure. Anther. Anther is bilobed and each lobe in it is diathecous, means it contains two theca. It is a bilobed structure and consists of two theca. It is a tetragonal structure with four microsporangia. This structure is a tetragonal structure and it contains four microsporangia, two at each corners. And these theca are separated by a longitudinal groove running lengthwise. Microsporangia are located at the corners. They further develop to form pollen sacs which contain pollen grains. Microsporangia develop into pollen grains. Let's go on a blank slide. Yeah. Nature of anther. As you know, anther is angiospermic. I am making points so that we will remember these points about the anther. It is a bilobed structure. It is tetragonal. Diathecous means contains two theca. And it is tetra microsporangiate. Right? Flower. It is the fascinating organ of angiosperms. It consists of calyx, corolla, androecium and gynoecium. It is a transverse section. A typical microsporangium appears near circular in outline, means its outer structure is a circle. It is surrounded by four wall layers: epidermis, endothecium, middle layer, and tapetum. The outer layers are protective and help in dehiscence of anther, and the tapetum provides nourishment to the developing pollen grains. It has four wall layers. Microsporangium has four wall layers, right? Epidermis, endothecium, middle layers, and tapetum. These three layers are the outer layers and are protective. They develop into wall, anther wall, whereas tapetum provides nourishment to the developing pollen grains. And it develops into sporogenous tissue. Cells of tapetum possesses dense cytoplasm and generally contain more than one nucleus. This dense cytoplasm gives nourishment to the pollen grains. In young anther, the sporogenous tissue, that is the compactly arranged homogeneous cells which are present at the center of each microsporangium, leads to the development of pollen grains. Firstly, when sporogenous tissue is young, at young stage, MMC, that is the mother cell, divides into microspore, develops into microspore, and from microspore, pollen grains are developed. And when pollen grains mature, they lead to the dehiscence of anther. Microsporogenesis. It is the process of formation of microspore from PMC, that is the pollen mother cell, through meiosis. The process of formation of microspore from PMC is known as microsporogenesis. As the development occurs in anther, the sporogenous tissue undergoes meiosis to form microspore tetrad, and hence each cell is a potential pollen or mother cell. Sporogenous tissue undergoes meiosis to form microspore tetrad. In this, sporogenous tissue undergoes meiosis and forms microspore tetrad, and from this potential pollen or mature pollen grain develop. Each cell of sporogenous tissue has capacity to give rise to a tetrad. As the anther matures, the microspores and dehydrates get detached from each other and develop into pollen grains. As the anther or pollen grains mature, they get detached from each other and develop into pollen grains. Thousands of pollen grains formed inside microsporangium released with the dehiscence of anther. Here you can see the process of microsporogenesis. Firstly, sporogenous tissue undergoes meiosis and forms microspore tetrad. And when microspore tetrad undergoes further meiosis, it forms microspore, that is the mature one. And as the anther matures and dehydrates get detached from each other, it develops into pollen grains. And when pollen grains mature, they are released from microspores. Here you can see the diagram. This layer is known as middle layer. Here the innermost layer is known as tapetum. Inside tapetum, this tissue, white color fluid, is known as sporogenous tissue. This layer is known as endothecium. This outermost layer is known as epidermis. epidermis, endothecium and middle layer. These are the protective layers, whereas tapetum provides nourishment, and this is known as connective. Here you can also say epidermis, endothecium and middle layers, protective layers, and this is tapetum. Here you can see dehiscence of anther leads to the release of pollen grains. Pollen grains represent male gametophyte and is spherical in shape with about 25 to 50 micrometers in diameter. They are about 25 to 50 micrometers in diameter. They have two wall layer, exine and intine. They have two wall layers, exine and intine. Exine is the outer layer and intine is the inner layer. Exine is hard layer which is made up of sporopollenin, whereas intine is the thin layer and continuous layer which is made up of cellulose and pectin. It is made up of sporopollenin and it is made up of cellulose and pectin, which is sporopollenin is an extremely resistant organic material which can withstand high temperatures, high acidic and alkaline conditions and enzymes. Pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains exine, they have some apertures which are called germ pores. Their sporopollenin, that is the resistant material, is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. Only because of sporopollenin, pollen grains are preserved as fossils. Let me show you the difference between exine and intine so that you can remember. Exine is the outer layer, intine is the inner layer, right? It is developed it contains sporopollenin and it contains cellulose and pectin. It is a hard layer, it is a thin layer, it is a continuous layer, whereas it is discontinuous. And sporopollenin is absent in germ pores. A plasma membrane surrounds the cytoplasm of pollen grains. Mature pollen grains, when pollen grains mature, they contain two cells, vegetative cell and the other cell is known as generative cell. Let's see the difference. Vegetative cell and generative cell. Vegetative cell is bigger, whereas generative cell is small. As you can compare this in the diagram, that vegetative cell is bigger in size, whereas generative cell is small. Vegetative cell has abundant food reserve, whereas generative cell floats in the cytoplasm of vegetative cell. Vegetative cell has large irregular shaped nucleus, whereas generative cell has spindle shaped nucleus with the dense cytoplasm. In more than 60% of angiosperm, pollen grains are shed at which stage? They are shed at two-celled stage. And remaining angiosperms, generative cell divides mitotically further to and give rise to two male gametes before pollen grains are shed at three-celled stage. More than 60% angiosperms, pollen grains are shed at two-celled stage, whereas in remaining angiosperms, generative cell divides mitotically and pollen grains are shed at three-celled stage. Here you can see enlarged view of pollen grain tetrad and stages of microspore maturing into pollen grain. Here you can see pollen grain tetrad enlarged view. Here you can see all other diagrams of vegetative cell, generative cell, and first before they develop into vegetative and generative cell, they form asymmetric spindle. Effect of pollen grains on humans. Pollen grains affect humans in many ways. Allergy. It causes allergy and bronchial afflictions. The person may be get sneezing or may start coughing when exposed to pollen grains. It may lead to chronic respiratory disorders also, like asthma, bronchitis, etc. These are the diseases that are the respiratory diseases caused due to pollen grains. Example, Parthenium, that is the carrot grass. Pollen products. They are rich in nutrients, and pollen tablets and syrups are used by athletes to increase their performance. They are banned now, but they are used in the past times. Period of viability. Once shed, the pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. It means that if we want to bring about fertilization or the plant wants to bring about fertilization, pollen grains have to land on stigma. So to bring about pollination, otherwise they will lose viability. Period of viability depends upon two conditions, temperature and humidity. It depends upon two conditions, that is the temperature and another one is the humidity. Example, cereals, pollen grains. They lose viability within 30 minutes, but some have the viability for months, like members of Rosacea, Leguminaceae, and Solanaceae. It is possible to store pollen grains of a large number of species for years in liquid nitrogen. If we store pollen grains in liquid nitrogen, such preservation of pollen grains is known as cryopreservation. Preservation means preserve some material, and if we preserve pollen grains or store pollen grains for large number of species for years in liquid nitrogen, it is known as cryopreservation. Such stored pollen grains can be used as pollen banks. We can use it as pollen banks similar to seed banks in crop breeding programs. Gynoecium, that is the female gametophyte, and formation of female gametophyte, that how it's develop. Gynoecium represents female reproductive part. It may be monocarpellary, that it may contain one pistil, or it may be multicarpellary, means contains more than one pistil. Pistil is stigma, style, and ovary. They are together known as pistil. The pistil may be fused. If they are fused, they are known as syncarpus, or if they are free, they are known as apocarpus. Let's go on a blank slide. Let me show it with the help in the diagrammatic way. Yeah. Gynoecium, as you know, it is the female part. It may be monocarpellary, or it may be multicarpellary, means contain more than one pistils. If it is multicarpellary, it may be syncarpus or apocarpus. Syncarpus means pistils are fused into one, and apocarpus means they are free. Infused, its example is in papaver plant, and apocarpus example is rose. It contains many pistils, so it is, and that pistils are free. Each pistil consists of stigma that receives the pollen grains, or style, that is the elongated cylinder part below stigma. Ovary, that is the bulged basal part containing the placenta, or we can say ovules, which is located inside the ovarian locule, that is the cavity. The placenta consists of megasporangia or ovules. The number of ovules in ovary may be 1. In the case of wheat, paddy, and mango, the ovules number is 1. But there are many in case of papaya, watermelon, and orchids. You can compare it with the help of seeds, like in mango. It is one seeded, whereas watermelon contains many seeds. Here you can see the diagram, stigma, style, and here ovary, and below ovary this part is known as thalamus. This is syncarpus, means fused, right? Here you can see the structure of a ovule. This part, upper part, is known as hilum. This is known as funicle. This gap is known as micropyle end, and at the opposite of micropyle end, this is the chalazal point. This is embryo sac, and outside embryo sac, this is nucellus. Inner integument, that is the protective layers, and this is the outer integument, and this end is known as micropylar region. This is the micropyle entering region, and this is the micropylar region, and out at the opposite side of micropylar end, this is the chalazal point. Megasporangium. Ovule is a small structure. It is attached to placenta by funicle or a stalk. Ovule is a structure which is attached with the help of a stalk known as funicle. The junction where ovule and funicle meet are known as hilum. Here you can see in the diagram. Funicle and ovule where meet, that junction is known as hilum. Each ovule has one or two protective layers called integuments, outer integument or inner integument, that covers the rest of the ovule except for a small opening that is known as micropyle, and the chalaza lies on the opposite of micropyle, and that represents the basal part of ovary. Nucellus is present within the integuments, and it contain reserved food for the developing pollen grain. For the developing embryo, and nucellus provides all the food which is needed for its development. The embryo sac is located within the nucellus. We call embryo sac as female gametophyte also. Megasporogenesis. It is the formation of megaspores from megaspore mother cell. Means megaspore mother cell develops into megaspores. This process is known as megasporogenesis. Ovules generally differentiate a single megaspore mother cell in a micropylar region of the nucellus. The megaspore mother cell is large and it contains a dense cytoplasm and a prominent nucleus. Here you can see that if the question arises that what a megaspore mother cell contains or how it looks, it's large or small. It is large and it contains a dense cytoplasm and a prominent nucleus. It undergoes meiosis to form 4 megaspores. Out of 4, it contains MMC contains 4 megaspores, out of which 4 out of 4 only 1 is functional and forms gametophyte. Other 3 degenerate. Here you can see the process of megasporogenesis with the help of this diagram. Let's start it. One of the cell of the ovule in the micropylar end firstly differentiate into megaspore mother cell, and when megaspore mother cell undergoes meiosis, or that process is known as megasporogenesis, 4 megaspores are formed, out of which 3 generate and 1 is functional, and that develops into embryo sac or female gametophyte. An ovule generally has a single embryo sac. Monosporic development, that embryo sac formation from a single megaspore. If embryo sac is formed from a single megaspore, then it is known as monosporic development. Here you can see the diagram. This is the micropylar end, this is nucellus, and this is megaspore mother cell. Pollination. It is the process of transfer of pollen grains from anther to the stigma of a pistil. This is the definition for pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. Pollination is of three types. Let's go on a blank slide. Yeah. Pollination is of three types. Autogamy, geitonogamy, and xenogamy. In autogamy, it involves self-pollination. Means pollination occurs between same plant and same flower. In geitonogamy, it is genetically self-pollination. But economically, it involves cross-pollination. Cross-pollination means different plant and different flower. In xenogamy, only cross-pollination occurs. Means between different plant and different flower. Here you can see the difference between xenogamy, autogamy, and geitonogamy. In autogamy, there are two types of flowers: cosmogamous flowers and clistogamous flowers. Cosmogamous flowers are those which have exposed anther and stigma. Means anther and stigma are very much visible. They are exposed. But in clistogamous flowers, they don't open at all and only autogamy occurs in that flowers. Now you know that there is two types of plants. One is bisexual, other one is sexual. Bisexual plant is known as monoecious plant, whereas sexual is known as monoecious. In monoecious plant, autogamy, xenogamy, and geitonogamy. Any one can occur, whereas in monoecious plant only geitonogamy and xenogamy can occur. Agents of pollination. Plants use air, water, abiotic. Air and water are the abiotic agents, whereas animals are the biotic agents which are used for pollination by plants. Majority of plants use biotic agents for pollination, only a small portion use abiotic agents. Pollen grains coming in contact with stigma is a chance factor in both wind and water pollination, that if they come in contact with the stigma, there will be pollination. Otherwise, there is no chance of pollination to occur in case of wind and water. Pollination by wind. It is the most common form of abiotic pollination. Plants possess well-exposed stamens and large feathery stigma, so that pollen grains should be light and non-sticky, so that they can be easily carried away by winds. And if that pollen grains come in contact with stigma, they bring about fertilization. Otherwise, no pollination will occur. Pollens should be light and non-sticky in case of wind pollination. Wind pollinated flowers often have a single ovule in ovary and have numerous flowers that are packed in inflorescence. Example, cotton and date palm. It is also common in grass. Pollination by wind is common in grass. Pollination by water. Pollination by water is quite rare in flowering plants. flowering plants and is limited to about 30 genera, mostly monocotyledons, means pollination by water is rare and it occurs mostly in monocotyledons. As against this, water is a regular mode of transport. It is believed, particularly from some bryophytes and pterophytes, that their distribution is limited because of the need of water for transport of male gametes and fertilization. Their distribution is limited because they need water for their transport. Some examples of water pollinated flowers are Vallisneria and Hydrilla, which grow in fresh water, and several marine seagrasses such as Zostera. If they can tell or give you a question that give an example of seagrass that is pollinated by water, that is Zostera, and flowers that bring about water pollination is Vallisneria and Hydrilla. Not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. In plants like water hyacinth and water lily, flowers are above the level of water, so they are pollinated by either insect or wind. In Vallisneria, the female flower reach the surface of water by a long stalk, and the male flowers or pollen grains are released onto the surface of the water, and so they are carried by water currents to bring about pollination. In another group of water pollinated plants, such as seagrasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribbon like, so they can be easily carried away by water. Some of them reach the stigma and achieve pollination, and some may lose their viability. In most of the water pollinated species, the pollen grains, so they don't become wet, are covered by a protective layer known as mucilaginous covering. Both water and wind pollinated flowers are not colorful and do not produce nectar because they are pollinated either by water or wind. But if they are pollinated by insect, they may be colorful. Pollination by animals. Majority of flowering plants use a range of animals as pollinating agents, butterflies, bees, etc. These are the common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Most of the insect pollinated flowers are large, they are colorful, fragrant, and contain nectar to attract the animal pollinators. These are their floral rewards. Floral reward can be in the form of providing safe places to lay eggs. Nectar and pollen grains. They are the usual pollen floral rewards. All symbiotic relationship exist between the plant, yucca, and its pollinator moth. The moth is dependent on the plant because it deposits its egg on the licuole of the ovary of the plant, and in turn the plant is pollinated by moth. So they are interdependent on each other. The pollen grains are sticky and get stuck to the body of the pollinator. Because of their stickiness, they get attached to the feet of the pollinator. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Repeated self-pollination leads to the inbreeding depression. If there may be repeated self-pollination may occur several times, it may lead to inbreeding depression, and the plants have developed to prevent such self-pollination. We can Autogamy is prevented by following ways. Autogamy is prevented as pollen release and stigma receptivity. They are not coordinated. If pollen releases, then stigma will be not receptive, and different positioning of anther and stigma. Self incompatibility. This is a genetic mechanism and prevents self pollen from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in pistil. Self incompatibility that prevents self pollination. Production of unisexual flowers. Either female or male flowers production may also leads to or prevent self-pollination. Presence of both male and female flowers on the same plant, such as castor and maize, they prevent autogamy. Outbreeding devices, ways to prevent both autogamy also and geitonogamy also. Presence of male and female flowers on different plants such that each plant is either male or female. This will prevent autogamy and geitonogamy. This mechanism is present in the case of papaya. Pollen pistil interactions. All events from deposition of pollen on stigma till the pollen tube enters the ovules. This is known as pollen pistil interaction. This is the definition for pollen pistil interaction. Pollination doesn't always ensure that the transfer of pollen grains will be compatible pollen grains. They may be incompatible. Hence, pistil has the ability to recognize the right type of pollen to promote post-pollination events. Pistil has that capacity that it may recognize the right type of pollen to promote post-pollination events. If the pollen is of wrong type, it will prevent pollen germination. The interaction is mediated by chemical components of pollen and pistil. The interaction is dependent on chemical components. Pollen pistil interaction is a dynamic process. It involves pollen recognition. Firstly, the pollens are recognized. Then, if the pollens are okay and is of right type, they followed by promotion, and if they are of wrong type, there will be inhibition of the pollen. The pollen tube then reaches the ovary, enters the ovule through micropyle, and then through filiform apparatus it reaches the synergids. Pollen tube then reaches the ovary and enters the ovule with the help of micropyle, and this through filiform apparatus it reaches synergids. In this way, pollen tube grows. Compatible pollination steps. Pistil accepts the pollen, then formation of pollen tube occurs, then pollen tube reaches ovary, pollen grains move into pollen tube, then pollen tube enters into ovules through micropyle, then pollen tube enters filiform apparatus and fertilization occurs. And if there will be incompatible pollen, then pistil will reject the pollen, prevents pollen tube growth and prevents fertilization. Here you can see the diagram. Artificial hybridization. It is a method to improve crop yield. Farmers mostly use artificial hybridization because it is a method to improve crop yield. In this method, it is essential to ensure that the right kinds of pollen grains are used and the stigma is protected from unwanted pollen grains. And if we want to protect the stigma from unwanted pollen grains, it is achieved by two processes: emasculation or bagging. Emasculation is the process in which the anther is removed from the bud if the female parent bears bisexual flowers, and it prevents self pollination and takes place before anther matures. And in case of bagging, the emasculated flower is covered by a bag so as not to allow contamination of the stigma by unwanted pollen grains. When the stigma of the bagged flower becomes receptive, collected pollen grains are dusted and then the flower is again rebagged to bring about fertilization. If the female parent is unisexual, emasculation is not this necessary. In this case, female bud is directly bagged, and when the stigma turns receptive, suitable pollen grains are dusted onto it so to allow germination. Double fertilization. When the pollen grains fall on the stigma, the pollen tube enters into one of the synergids and releases two male gametes. When pollen grains fall into stigma, then pollen tube enters one of the synergids and then releases two male gametes. One of the male gamete moves towards the egg cell and fuses with it to complete the sign gamete to form the zygote. One of the male gamete moves towards the egg and fuses with it to form zygote. The other male gamete fuses with the two polar nuclei and forms triploid primary endosperm nucleus. One forms zygote and other forms triploid primary endosperm nucleus. This is known as triple fusion. Since two kinds of fusion takes place, sign gamete, that is the formation of zygote, and triple fusion, that is the formation of triploid primary endosperm nucleus. That's why this fertilization is known as double fertilization. After triple fusion, the central cell becomes primary endosperm cell. The primary endosperm cell nucleus gives rise to the endosperm while the zygote develops into embryo. The primary endosperm nucleus firstly gives rise to the endosperm while the zygote develops into embryo. Post-fertilization events. Post-fertilization events means the events that takes place after fertilization, double fertilization. All events that occur in a flower after double fertilization are known as post-fertilization events. It includes development of endosperm and embryo and maturation of ovules into seeds and ovaries into fruits. Formation of endosperm. Let's see how the endosperm is formed. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. Firstly, the primary endosperm cell divides and forms triploid endosperm tissue. Then the cells of the tissue are filled with reserve food which are used for the nutrition of developing embryo. The primary endosperm nucleus repeatedly divides to give rise to free nuclei. This stage of development is called free nuclear endosperm. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut is nothing but free nuclear endosperm. As you can see, the coconut water which we get from the tender coconut, it is free nuclear endosperm that is made up of thousands of nuclei, and the surrounding white kernel that is known as cellular endosperm. Cell wall formation occurs next, and endosperm may be either fully consumed, as in pea or beans, or the portion of endosperm remains in the matured seed, as coconut and castor. You can see the difference of albuminous seed and non-albuminous seed. In albuminous seed, endosperm is not completely used by the developing embryo, and in non-albuminous seed, endosperm is completely used by the developing embryo. Its example is coconut, castor, and maize, and in non-albuminous its example is pea, bean, and mustard. Development of an embryo. The embryo develops at the micropilar end of the embryo sac where the zygote is situated. Where embryo develops, it develops at the micropilar end. Most zygotes divide only after certain amount of endosperm is formed. This is adaptation to provide assured nutrition to the developing embryo. This lesson is important, that most of the zygotes divide only after certain endosperm is formed. This is an adaptation. This is an important adaptation so that assured nutrient or nutrition is provided to the developing embryo. The zygote gives rise to the proembryo, then to globular, then heart-shaped and mature embryo. A typical dicot embryo consists of an embryonal axis and two cotyledons. A typical dicot embryo contains which things? An embryonal axis and two cotyledons. The portion of the embryonal axis above cotyledons is known as epicotyle and below is known as hypocotyle. It contains radicle, that is the root tip, and the root tip is further covered by root cap. Here you can see plumule. These are the two cotyledons. Hypocotyle, that is the upper portion. Lower is known as epicotyle. Here is the radicle and this is the root tip. The upper portion is known as epicotyle, whereas the lower is known as hypocotyle. In a monocot embryo, there is only one cotyledon, and like in grass, it is known as scutellum. Here you can see, in monocot, there will be one cotyledon, like in a grass, it's known as scutellum, and is situated at the one side of the embryonal axis, and at its lower end, the embryonal axis had the radicle or the root cap. Here you can see, it has the radicle or the root cap, and the lower end is known as coryza. The epicotyle lies above the scutellum and has the shoot apex. Leaf primordia is enclosed in hollow structures called calyptiles. In this, leaf primordia are enclosed in a hollow structure that is known as coryza. Development of seeds. How seeds are developed. It is the last stage of sexual reproduction in angiosperms. Seeds are fertilized ovules that develop inside a fruit. A seed contains these three things: seed coat, cotyledons, and embryonal axis. What is a seed coat? It is a double layered and formed by integuments. It contains two teguments. Integuments means layers. It contains two layers: testa, that is the outer layer, and tegumen, that is the inner layer. Seeds may be albuminous, that is the endosperm present, or maybe non-albuminous, means endosperm absent. Some seeds, such as black pepper and wheat, have remnants of nucellus known as perisperm. The integuments of ovules harden to form the seed coat, and the micropyle facilitates the entry of oxygen and water into the seed. Integuments of the ovules harden, that's why they form seed coat, and micropyle facilitates the entry of oxygen into the seed for its development. As it loses moisture, the seed may enter dormancy, or if favorable conditions exist, it germinates. Advantages of seed to plants. Seeds reserve food materials. Seed coat provides protection. Seeds of large number of species live for several years, and we get plants get more and more number increased. Seeds have better adaptive strategies for better survival, and to mankind they are used as food, or seed is the basis of agriculture. Without seed, there will be no food. Dormancy, it is a period in organism's life when growth, development, and physical activity are temporarily stopped. Means they are stopped for a temporary time, for a minimum time. Seed viability. The germination power of a seed has a time limit on expiry of which the seed will not be germinated. It will become a normal grain, that is known as seed viability. Dicotyledonous seed has a seed coat, hilum and micropyle, whereas on the other hand monocotyledonous seed has a seed coat which is fused with the pericarp and endosperm. Pericarp and endosperm are the layers of seed. Micropyle is a small opening on seed coat which facilitates the entry of oxygen and water, as we have discussed earlier. And hilum is a scar which is present on the seed coat. Development of fruits. How fruits are developed. Ovary of a flower develops into fruit. The ovules develop into seed, and ovary develops into fruit. The valves of ovary transform into valves of fruits that are known as pericarp. Fruits may be fleshy, as in mango or orange, or can be dry, as in groundnut and mustard. In some plants, floral parts other than ovary take part in fruit formation. In these, the thalamus contribute to fruit formation. Such fruits are known as false fruits. Fruits that develop from the ovary are known as true fruits. Fruits that are developed from ovary, development of fruits taken by from ovary, they are known as true fruits. But if fruits are developed without ovary, they are known as false fruits, and the false fruits examples are apple and strawberry. Example are apple and strawberry. And true fruits examples are tomato, grapes. Some fruits develop without fertilization, and they are known as parthenocarpic fruits. Like example, banana. Fruits that develop from ovary but without fertilization, they are known as parthenocarpic fruits. Its example is banana. Fruit structure. Wall of fruit are known as pericarp. Epicarp is the outer layer, and mesocarp is the middle layer of cells with varying thickness, and endocarp is the innermost layer. Pericarp fruits wall is known as pericarp. Epicarp is the outermost layer. Then middle layer of cells is known as mesocarp, and endocarp is known as the inner layer. Other modes of reproduction. Apomixis and Polyembryo. Some plants produce seeds without fertilization. The plants which produce seeds without fertilization, that process is known as apomixis. It is a form of asexual reproduction that is some time mimicking sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction, division, and develops into embryo without fertilization. Means the seeds produced without fertilization is known as apomixis. Apomixis is important for producing hybrid varieties of fruits and vegetables, and for increasing crop yield multifold. This is the advantage of apomixis. One of the problems of hybrids is that hybrid seeds have to produce every year. If the seeds collected from hybrids are sown, the plants in the progeny will segregate and don't maintain hybrid characters. Production of hybrid seed is very costly, and hence the cost of hybrid seeds become too expensive for the farmers to bring about hybrid seeds. If these hybrids are made into apomix, there is no segregation of characters in the hybrid progeny, then the farmers can keep on using the hybrid seeds to raise new crop year after year, and he doesn't have to buy hybrid seeds every year. If a farmer use apomixis process, then he doesn't need to buy hybrid seeds which are very costly for him. So he can use Apomixis to buy seeds and grow hybrid varieties year after year. Because of the importance of apomixis in hybrid seeds industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into hybrid varieties. And whereas occurrence of more than one embryo in a seed is referred to as polyembryo. If there will be seeds are produced without fertilization, that is known as apomixis, and if more than one embryo is present in a seed, then it is known as polyembryo. Please like, share and subscribe to my channel if you like my video. Thank you.